Biotechnology and Biofertilizers. What is biotechnology? Biotechnology is the use of living organisms, biological systems, or their components to develop products and processes for the benefit of humans, animals, plants, and the environment. What are biofertilizers? Biofertilizers are living microorganisms, such as bacteria, fungi, algae, that promote plant growth by increasing the availability of nutrients in the soil. They are an eco-friendly alternative to chemical fertilizers and are a key product of biotechnological innovations in agriculture. How biotechnology is used in biofertilizer production. Biotechnology helps isolate and identify efficient strains of microbes, example Rhizobium, Azotobacter, Pseudomonas. Genetically modify microorganisms for enhanced nutrient fixation and stress tolerance. Mass-produce biofertilizers using fermenters and optimized growth media. Formulate and package microbes in suitable carriers, example peat, lignite, liquid formulations. Monitor quality and survival rate of microbial strains. Major types of biofertilizers. Type. Example microorganisms. Function. Nitrogen fixing. Rhizobium, Azotobacter, Azospirillum, Anabina, Nostoc. Fix atmospheric nitrogen into usable form, NH4, NO3. Phosphate solubilizing. Bacillus megatherium, Pseudomonas striata, Aspergillus. Convert insoluble phosphate into soluble form. Potassium solubilizing. Frateria aurantia, Bacillus mucilaginosus. Release potassium from minerals. Mycorrhizal fungi. Glomus, Gigaspora. Enhance nutrient and water uptake. Cyanobacteria/BGA. Anabina, Nostoc, Tolipothrix. Fix nitrogen in paddy fields. Compost biofertilizers. Trichoderma, Cellulomonas. Decompose organic matter into humus. Biotechnological innovations in biofertilizers. Genetically engineered microbes, modified Rhizobium or Azotobacter strains for higher nitrogen fixation efficiency. Liquid biofertilizers, developed using biotechnology for better shelf life and higher cell count than solid forms. Microbial consortia, combination of different beneficial microbes, example Rhizobium plus PSB plus Azospirillum, to provide multi-nutrient support. Encapsulation technology, encapsulation of microbes in alginate beads or nanomaterials to enhance survival and release rate. Bioinformatics and genomics, genome sequencing of microbial strains to identify key genes responsible for nutrient fixation and stress tolerance. Importance and applications. Enhance soil fertility, improve natural nutrient cycles. Reduce chemical fertilizer use, lower cost and pollution. Promote sustainable agriculture, eco-friendly and renewable. Increase crop productivity, stimulate root and shoot growth. Improve soil structure, increase organic matter and microbial activity. Environmental protection, reduce nitrate leaching and greenhouse emissions. Mechanism of action. 1. Nitrogen fixation. Certain bacteria and algae convert atmospheric N2 into ammonia, NH3, or nitrates that plants can absorb. Example, Rhizobium forms nodules on legume roots. Rightward arrow enzyme nitrogenase catalyzes N2 rightward arrow NH3. 2. Phosphate solubilization. Microbes release organic acids that dissolve in soluble phosphate minerals, like tricalcium phosphate. 3. Production of growth hormones. Microbes produce auxins, gibberellins, and cytokinins. Rightward arrow stimulate root and shoot growth. 4. Biocontrol action. Some biofertilizers, example Trichoderma, Pseudomonas fluorescens, suppress soil pathogens and enhance plant defense. Advantages. Eco-friendly and renewable. Cost-effective. Improves soil health and structure. Enhances nutrient uptake. Supports organic farming. Reduces pollution from chemical fertilizers. Limitations. Sensitive to temperature, pH, and storage conditions. Short shelf life, especially solid formulations. Require proper application technique. May compete with native soil microbes. Slower visible results compared to chemical fertilizers. Future prospects. Genetically engineered microbial inoculants with multi-nutrient and stress-tolerant capabilities. Nanotechnology-based biofertilizers for controlled nutrient release. CRISPR-based genome editing to enhance nitrogen fixation genes. Integration with precision agriculture and remote sensing for soil microbiome monitoring. Biotechnology has revolutionized the development of biofertilizers, providing sustainable and eco-friendly alternatives to chemical fertilizers. By using advanced microbial engineering, mass cultivation, and nanotech-based formulations, biofertilizers are shaping the future of green agriculture and environmental sustainability.